Welcome to this lecture on asexual reproduction of bacteria and other prokaryotic organisms. In this particular lecture, I shall be discussing the asexual reproductive mode of different prokaryotic organisms like the myxobacteria, then the mycoplasma, then the rickettsia, chlamydia, spirocheta, and actinomycetes. This myxobacterial cell is having normal bacterial cell which looks like a typical rod-shaped or curved bacteria which will divide by binary fission. But sometimes it may be elongated, producing a filamentous structure, and not only that, it will produce a slimy secretion outside, and thereby it can exhibit a typical slime movement which is commonly found in slime mold or myxomycetes, and that is the reason why it is called myxobacteria. A very important point is that this particular bacteria will have a relatively larger genome size than normal bacteria, which may be almost 9 to 10 million nucleotide. So that way they are advanced over normal bacteria. So what happens is this typical filamentous structure will reproduce and will form a huge mound-like structure. You can see this is the mound-like structure produced by the filaments of myxobacteria. And as they grow in number, they will also take the nourishment from the substratum here, as a result of which there will be deficiency in the nutrient. And if they encounter this deficiency, they will move away from this particular nutrient deficient region. But the ones which are already there, they have to stay back. So what do? They actually produce a typical cylindrical structure with an apical swollen portion. And this swollen portion will give rise to minute finger-like projections. And these finger-like projections will then produce a swollen round or globose sporangium. And this sporangium is actually the myxosporangium. And this sporangium will develop the myxospores within, and with the advent of favorable condition, these myxospores will come out. So it is one strategy. These myxospores will ultimately help the organism to survive during unfavorable condition. And when the myxospores will germinate, they will again produce the normal bacterial cell which will reproduce by binary fission. And this is the actual photomicrograph of a myxosporangia, and that will liberate the myxospores. So in the life cycle of myxobacteria, we see mainly two types of reproduction. One is binary fission like normal bacteria, and the other one is the production of myxosporangia and myxospore, which has actually taken them very close to myxomycetes. Mycoplasma, unlike other bacteria, is devoid of any definite cell wall. It is not having any particular wall, so it is having variable shape. But there are some rod-like cells which will divide by binary fission. So this binary fission is the common way of reproduction in mycoplasma. And this binary fission is actually being followed by the DNA replication, where the DNA will replicate, and this replication will proceed from the DNA A in both the direction. Now normally the speed is relatively slower than other bacteria. So the mycoplasma DNA replication will occur very slowly. Not only that, it is progressing on both ends, but in one particular site it is arrested, it is stopped. And the moment it is arrested, you can see in this particular slide that it is getting branched. So this is the normal case of binary fission, where the nucleoid is getting divided and then it gets elongated and two rods are produced, as you can see here, actual electron micrograph. Here what happens is the DNA replication has been arrested, as a result of which the mycoplasma is getting branched, and thereby it will produce this multi-lobed structure. This DNA replication is actually seen here in the electron micrograph, where you can see that the replication is taking place and the cell is dividing into rod-like cells, or sometimes it is producing multi-lobed structure. The particular DNA replication is because of specific purpose. You can see in this particular slide that the gradual formation of gene from K to C is gradual, and thereby it will produce the specific gene product, where the C is producing the B, which is producing the adherence protein. So because this adherence protein is in the terminal finger-like projection of the mycoplasma, it will help the organism to adhere to the substratum, and thereby they can draw nourishment. At the same time, when they are producing another particular protein which is outside in the form of A, it is accounting for slimy movement. So in one way we can say it is the DNA replication which is helping in the movement of the mycoplasma. But remember, the mycoplasma is without cell wall, so it will not produce true spore. And this is an advantage. A normal antibiotic which is inhibiting cell wall in bacteria will have no action on mycoplasma. But this particular mycoplasma may act as super antigen. This super antigen is received by specific B cell receptor, and then it is going inside the cell and it will actually attack the mitochondria, and the mitochondria will produce the specific cytochrome oxidase, and thereby this particular enzyme will be activated, which is caspase 3 and caspase 9, and this caspase activation will result in apoptosis. So in one way what we see is, because of the super antigen or mycoplasma or any other infection acting as super antigen, the cell is ultimately getting destroyed. So this is an advantage for cancer cell, where this apoptosis will prevent cancer development or spread.
Sir, mycoplasma cells are also called PPLO. Why?
You see, I showed you in this particular slide that mycoplasma is having variable shape. They are not having specific shape. So they are called pleomorphic. At the same time, they can cause pneumonia. So the full form is pleuropneumonia-like organism. Okay? Rickettsia is a typical longitudinal bacteria-like cell which is dividing by transverse or longitudinal binary fission. And thereby this process of binary fission is also sometimes called fragmentation. So we say that they divide by both binary fission and fragmentation. And sometimes when they divide, they will produce a long trail of cell. This is actually the Rickettsia rods. Due to the longitudinal fragmentation, they have actually produced a long trail within the host cell. And they do not produce any definite spore, but they will be inside the vacuole of the cell, and thereby they are protected from the other host defensive enzymes. And thereby when they move, actually they are moving along with the vacuole. So this is an advantage of Rickettsia. And within that particular Rickettsia cell, we can see different types of morphology. There may be large spherical initial, there may be medium spherical initial, which will be having around 1 to 2 microns, sometimes very large. But there may be at the same time the elementary bodies which will be very small, maybe into 500 to 600 nanometer, or sometimes we call it spherical initial, which is slightly larger than the elementary body. But all these different shape and size of this particular Rickettsia cell will help them to survive within the host. Depending upon the nature of the defense of the host, they will survive in different forms. So they are actually showing pleomorphic activity within the host cell.
Sir, what is the function of the elementary bodies that are formed during replication in Rickettsia?
You see, elementary bodies, they are not true cells. I told you they do not produce true spore, but here elementary body will act as a spore. It will act as a resting spore which will continue to stay within the host body and will not be attacked by any defensive enzymes. Chlamydia is actually divided into two types of cellular structure. One is called the elementary body, other one is called the reticulate body. You can see in this particular slide, the larger cells, they are called the reticulate body, and the smaller cells, they are called the elementary body, which is very close to that of the Rickettsia elementary body. Now this particular cells which are reticulate body, they are definitely large size, their nucleoid is clearly visible, and they are maybe in the order of 1.5 to 2 microns, sometimes very large. But elementary body, on the other hand, they look like spore. They are very small. They are have say 0.2 to 0.3 micron in diameter, and they actually act as infectious organism. So this particular elementary body is protected inside the cell by the host vacuole, where the enzymes will not reach. And the maturation of the elementary body is relatively faster. The reticulate body formation is relatively slow. So what we say is that the elementary body are relatively more, and when there is some host defense, the production of elementary body will further increase. But the reticulate body will be there only when the host is actually showing less defense reaction. The cell cycle of chlamydia clearly exhibit this type of differentiation between the elementary and the reticulate body. You can see this is how the elementary body is getting attached. It is actually received by the cell by the vacuolation. It is going inside the vacuole. Then it is showing replication inside the vacuole. And you can see the time lag is around 24 hours. And then gradually the vacuolation increases. So what happens is the vacuolation is almost occupying the entire cell. And as they are enlarging in size, they will produce the reticulate bodies at the same time. So these reticulate bodies will mature at the later stage when the elementary body is gradually getting high in number. And then ultimately what happens, the cell will undergo lysis and they will be releasing those elementary bodies, and these bodies will be again attached to specific cell receptor and they will enter into a fresh cell and causing the cycle again. So this is how this replication cycle of chlamydia is maintained between elementary body and the reticulate body. And this is important, as I was telling you, that the elementary body directly can actually attach to the host cell. You can see in this particular slide that they do have actual needle-like projection, which is a typical type 3 secretory needle or secretory projection, which will help in direct entry of the elementary body into the host cell wherever chlamydia is attacking. And chlamydia also can cause infection like chlamydia causing trachomatis or causing pneumonia-like symptoms. In this particular organism, you can see it is a typical spiral structure, and this spiral structure is not having any projection, but they are showing locomotion with the help of endoflagellum. And this is the spirocheta which is showing replication. You can see this particular spiral structure is getting replicated by binary fission. So this is the typical way of reproduction in case of spirochete. But along with it, you can see in this particular slide, there are some typical spore-like structure. So these spores are similar to endospores of bacteria. A particular spirocheta, as in this case is Borrelia, there will be different types of cellular morphology. One is the typical spiral morphology, one will be the spore-like morphology, and the other one will be the wall-less cell. So a typical Borrelia will develop into three types of structure. One is the spiral structure, which is looking like a corkscrew, and literally it can drill inside the cell and allow the infection of that cell. The second one is the spore, which is typically bacterial endospore having exosporium, mesosporium and endosporium, that is three-layered wall. There will be mesosome, there will be ribosome, all the other particulates will be there. But as the wall is getting thicker, the organelles will shrink towards the center. And the third form is the wall-less spirochete form.
Sir, does Borrelia attain all the three forms during its life cycle?
Yeah, it can, because I told you it can be normally a spiral shaped structure, but when inside the particular host, it will have host enzyme attacking it, so it will develop a spore. And sometimes when the spore is germinating, it will have the wall-less condition, and then it will ultimately elongate to produce a spiral again. So my answer is that the Borrelia will exhibit all the forms in its life cycle depending upon the stage of the life cycle and depending upon the external environmental condition in which the cell is surviving. Clear? This group of organism is in between bacteria and fungi. So like bacteria, it will have a nucleoid, that is the prokaryotic structure, and like fungi, it will have a typical mycelial structure, and this mycelia will develop a branch structure, and this branch will break, and thereby this fragmentation will occur, and this broken filament will produce a new plant body. But apart from that, it will also produce some typical rounded spore-like structure which are cut off in chains. Now these chains are cut off both along the surface of the ground or subterranean, or it can be aerial. And this type of spores are called conidia or conidiospores. So reproduction in case of actinomycetes occur mainly by fragmentation and also maturation of the conidia or conidiospores. This is reflected clearly in the life cycle of Streptomyces. This group of organisms, the life cycle starts with the spore. The spore will germinate to produce this filamentous structure, and then this filamentous structure will develop into a vegetative mycelia or mesh, and then they will produce aerial branches, and then from these aerial branches, these are the actual spore or conidiospores which are cut off, and this is how the life cycle is completed. Now this group of organism actually, though it is producing only conidia and only by fragmentation they are reproducing, but on the basis of their cell wall composition and sporulation form, they can be divided into 5 different types. The type 1 is this one which the cell wall is devoid of typical nitrogenous phospholipid. These are the different forms of type 1 actinomycetes, where in Streptomyces you will have the typical spiral development at the tip of the mycelia producing the spore. And sometimes this conidia in Streptoverticillium is produced in an umbel-like situation. You know, umbel meaning it will have an aggregate. And sometimes in Actinosporangium it will produce spores which are dropping off from the mycelia. So they are called drop spore. And sometimes they may develop an aggregate called sclerotia. Sclerotia is found in Chinea. Similarly, Elytrosporangium will develop intermediate swollen structure which are commonly called merosporangia. And sometimes the sporangia will have a single spore, as it is seen in case of Kita sativa.
Sir, what is the difference between true sporangia and merosporangia?
You see, the merosporangia, actually the literal meaning it is partial sporangium, but here they are producing terminal projection. So merosporangium is actually conidia-like structure or a conidiospore-like structure which will develop into chain of new conidia, unlike the sporangium of higher fungi. Type 2 is having specific cell wall where the phosphatidyl ethanol amine is present as the nitrogenous source in the phospholipid of the cell wall. Single apical conidia, the name itself suggests the genus, that is the Micromonospora, a single spore representing the conidia. Globose to laginiform sporangia is found in actinoplane. You can see these are the globose ones and these are slightly elongated, which are called laginiforms. Similarly, in case of Ampullariella, which is another type 2 actinomycete, there we will be having motile rod-shaped spores. These are the motile rod-shaped spores. That means the spores after being released from the sporangia will be showing motility. In Dactylosporangium, dactylo meaning finger-like projection arising from the mycelial mesh. Now type 3 is a very large group. Type 3 will have 3 subgroups, which is type 3A, 3B and 3C. But broadly, this is having a specific phosphatidylcholine without any characteristic sugar. This is the structure of the cell wall. The cell wall is having some relationship on the reproductive pattern. In type 3A, we have typical actinocinema. Actino meaning filamentous structure, cinema meaning aggregate. So actinocinema is having filamentous aggregate. And sometimes, as in case of Nocardiopsis, where you have long chain of conidia, the mycelia is being replaced by long chain which has produced a typical spiral-like network. Coming to the next group, which is commonly called the Geodermatophilus. In Geodermatophilus, what is there? The motile coccoid conidia. Coccoid meaning they're small rounded shapes, and these conidia, they are produced along the surface of the ground and they show motility. In the next group, which is Thermoactinomyces, the name itself suggests they can tolerate heat. So the cell wall is very hard. So Thermoactinomyces, they are having heat resistant endospore. And this heat resistant endospore actually represent a single conidia, and thereby they survive at high temperature. Type 3B is the second type, where we have short chains of conidia in actinomadura. Now this name itself suggests that 3B is not similar to 3A. So the cell wall is having madurose, which is a typical sugar component, which is different from 3A. So here in case of actinomadura, you can see the conidia, which is elongated, the lower part is oblong, the upper part is oval. But when we come to microbispora, there you can see typically two spore on two sides of the mycelia. That's why it's called micro, very small, bispora. And this is a typical paired conidia of microbispora. But when we come to the next one, which is called dermatophilus, dermat meaning skin and phili meaning love. So they are skin invading organisms. So this dermatophilus, mycelia and conidial mesh will invade the horse, and this is a typical dermatophilus infection in the horse's skin. The second set of 3B group include the microtetraspora. As the name signifies, you can see 1, 2, 3, 4 spore are formed on the typical finger-like projection. So this is called fourth cell sporangia of microtetraspora. We have the cylindrical two-spored sporangia of planobispora. In one plane, the two sporangia is dividing. Similarly, we have single cylindrical sporangia of planomonospora. So this was bispora because two spore. Here there is a single. This is a typical single reniform sporangia of planomonospora. But when we come to streptosporangia, strepto meaning in chain, so they are produced in long chain and long chain of streptosporangia, and there may be similarity with other group of organisms, but here the cell wall pattern is typical in case of streptosporangia, having, I told you, madurose sugar. Now this is the type 3C. Now this type 3C is having rhamnose and galactose in the cell wall, and this particular high sugar content of the wall actually give them the name Saccharothrics. So Saccharothrics is a group of organism having a typical coiled mycelia where the conidia is also embedded within the mycelial mesh. So you cannot see distinct projection. You can see of course here in the electron micrograph, but broadly under a normal microscope, this type of conidia is not visible. Now we come to the type 4. In this type 4, there is phospholipid in the wall having phosphatidylethanolammine, and at the same time there is glucosamine. So this type 4 is having a polysporous condition in actinopolyspora, wherever in case of micropolyspora, only the size of the spores are different and they are produced at any point in the mycelial structure. And when we come to the pseudonocardia, there will be both long conidia and short coccoid branches. So they are bimorphic and they are actually representing the pseudonocardia plant body. This is continuity of the type 4. You can see this dense mass is having discrete spore, discrete network of spore in Saccharomonospora, but these spores are distinctly observed. But when these spores aggregate, they will produce Saccharopolyspora. So just by seeing the spore morphology, the genera have been named. Coming to the last type, which is the long chain of conidia in Kitasatosporia. In Kitasatosporia, you can see this conidia is in long chains, and sometimes they are hanging from the branches because the branches are aerial and from where the long chains are hanging like stick. In conclusion, what we have learnt in this lecture is the asexual mode of reproduction in different bacteria-like organisms. We have started with Myxobacteria, then we have discussed Mycoplasma, Rickettsia, then Chlamydia, Spirocheta, and then actinomycetes. And in actinomycetes, we have learnt about the different cell wall form and how they have been classified on the basis of their reproductive spore. And on the basis of that reproductive spore, how the names of genera is deviating. So this is all about the asexual reproduction in bacteria and different other prokaryotic organisms.